Hi everyone, I'm Zixiachen from CSBL. The title of my presentation today is Autonomous Bioluminescence System. As we know, traditional biological imaging often faces limitations. Fluorescence requires external excitation, leading to background noise and phototoxicity, while traditional bioluminescence relies on exogenous substrate injection, making continuous monitoring difficult. Autonomous bioluminescence systems, which allow organisms to produce light entirely through their own metabolism, acting like living light bulbs. My report will combine the latest 2025 review paper and a classic 2020 biochemical study on key fungal enzymes. First, we must define what an autonomous luminescence system is. Unlike traditional methods, this is a genetically encoded platform that ingeniously integrates both the luciferase and the substrate biosynthesis pathway directly inside the cell. The advantages are immense. We no longer need to inject expensive substrate or worry about supply interruptions. This enables real-time, non-invasive, and long-term monitoring of biological processes without phototoxicity. Currently, the two most thoroughly characterized and widely applied systems are the bacterial lux system from prokaryotes and the fungal lux system from the eukaryotes. These two systems were compared using this table. The bacterial lux system has the longest history. Its core advantage is being fully genetically encoded. As early as 2018, enhanced operands were developed, and in 2025, a major breakthrough was achieved with the creation of the first autonomous transgenic mouse. However, lux faces challenges, especially a low quantum yield of around 0.1 to 0.2. In contrast, the fungal lux system is a newer discovery. Its highlight is exceptional performance in plants, achieving a naked eye invisibility. This is because plants naturally contain the fungal precursor caffeic acid. However, in non-plant hosts like animal cells, we must introduce additional metabolic pathway to synthesize caffeic acid, which increases the metabolic burden. The bacterial system is shown as Figure B. This is a cone light emitting reaction. The LuxAB enzyme utilizes reduced flavin, a long-chain fatty aldehyde, and oxygen to release blue-green light. But the key to autonomy lies in substrate recycling. Look at the Figure D. The light reaction consumes the aldehyde and produces a fatty acid. To sustain light, bacteria involved a fatty acid reductase complex composed of LuxC, LuxD, and LuxC. This complex consumes ATP and NADPH to reduce the fatty acid back to the aldehyde. This creates a perfect metabolic closed loop. As long as the cell provides energy, the light continues. Figure 2a shows how bacterial luciferase works at the atomic level. LuxAB is a heterodimeric protein. You can see that LuxA and LuxB share a highly similar TIM barrel fold. However, they are functionally distinct. Only LuxA possesses catalytic activity, and the yellow stick in the figure presents the FMN bond at the active site, and the LuxB is essential for stabilizing the structure of the LuxA. Without LuxB, LuxA is unstable and non-functional. This suggests that when performing genetic engineering, we must consider the correct folding and assembling efficiency of the both subunits. Next is about fungal system. The starting substrate here is caffeic acid. The cycle is driven by 4 key enzymes. First, hispase converts caffeic acid into hispidin. Next, H3H hydrolyzes hispidin to generate the actual luciferin, 3-hydroxyhispidin. Then, Luz oxidizes the luciferin to emit light. Finally, CpH recycled the product back to caffeic acid. To deeply understand the regulation of this system, we have selected the critical luciferin synthesis step catalyzed by H3H for detailed analysis based on a deep dive biochemistry study from 2020. H3H enzyme from the bioluminescent fungal Mycena chlorophyll, referred to as MCH3H. As shown in this figure, MCH3H is an NADPH-dependent FAD-containing monooxygenase. Its core function is to precisely hydroxylate the C3 position of the hispidin in the presence of oxygen, generating 3-hydroxyhispidin. This is a final preparatory step that ignites fungal bioluminescence. Researchers expressed and purified this enzyme in E. coli and found it to be a monomeric protein, which is distinct from the complex heterodimeric structure of the bacterial system. Researchers first characterized the basic properties of MCH3H. Look at the figure 1 on the left. MCH3H displays a typical flavoprotein absorption spectrum with characteristic peaks at 375 nanometer and 450 nanometer, confirming it binds a FAD cofactor. Even more interesting is figure 2b on the right, showing thermal stability experiments. The data shows that without substrate, the melting temperature is about 42 Celsius. However, the crucial finding is that upon adding the substrate hispidin, the Tm significantly rises to 50 Celsius. This indicates that substrate binding induces conformational changes that stabilize the enzyme, which is a classic substrate-induced stabilization mechanism. This slide presents the most important kinetic finding of the study. Compared Figure 5a and Figure 5b, these curves monitor the rate at which NADPH reduces the FAD cofactor. In Figure 5a, without the substrate hispidin, the reduction rate is extremely slow. However, in Figure 5b, once the substrate is added, the reduction rate is increased to 220 per second, an increase of nearly 100-fold. This reveals a sophisticated gating mechanism in MCH3H. The enzyme only allows NADPH to transfer electrons rapidly when the substrate is bound and ready. This effectively prevents the waste of NADPH and minimizes the production of the reactive oxygen species, thereby protecting the cell. Beyond understanding the mechanism, the next question is whether we can engineer it. Wild-type MCH3H prefers NADPH, but NADPH is generally more expensive and scarce in cells compared to NADH. Through structure modeling, researchers identified phenylalanine 44 and mutated into the glutamate. Please look at the highlighted data in Table 1. The F44E mutate shows a dramatically improved affinity for NADH, with the Km dropping from 240 micromole to 22 micromole. This means that with a single point mutation, it was successfully reversed the cofactor specificity, making the enzyme efficient at using NADH. This provides a vital tool for constructing synthetic biological systems with lower metabolic burden. So, how do they change the color of the light? Figure 5 illustrates two strategies. The first is about bacterial system. The most major strategy here is BRET. By fusing luciferase with fluorescent proteins of different colors, we can achieve a spectrum ranging from cyan to red, such as in the Nanolectern X series. The right panel shows the fungal system. Current strategies mainly rely on chemically modifying the hispidin substrate structure. However, this faces a major challenge. These modified substrates are difficult to synthesize in vivo using natural enzymes. Therefore, multicolor imaging in live fungal systems remains a bottleneck waiting to be broken. How do these systems perform in real-world applications? In Petunias, because the host naturally provides the substrate, we can see bright, naked-eye visible luminescence without any treatment. Meanwhile, the right panel displays a milestone achievement from 2025, the autonomous transgenic mouse. Although the brightness of the bacterial systems in mammals is not yet as high as in plants, it achieves true zero background, non-invasive, long-term imaging. This is revolutionary for tracking processes like tumor metastasis or development over extended periods. Beyond imaging, autonomous systems make excellent biosensors. As shown in Figure 7, we can design three types of probes. First is turn-off type, used for detecting toxin or antibiotics when cellular metabolism is damaged and ATP supply fails. The light rapidly dims. Second is the turn-on type, utilizing specific promoters so that the light pathway is achieved only when a specific signal, like heavy metal or pathogens, is detected. Third is the ration metric type, utilizing BRET principles to detect antilight like calcium concentration by changing the color of the light. This method cancelled out errors caused by variation in cell number or metabolic fluctuations. Despite immense progress, we still face challenges with low quantum yield and limited spectral ranges. Figure 8 depicts the roadmap for the future. We need to leverage the latest AI tools for de novo enzyme design. The goal of it is not just to patch existing enzymes, but to design superluciferases from scratch that are more compact, catalytically efficient, and capable of binding novel substrates. Furthermore, creating hybrid systems which combine the efficient substrate synthesis modules of bacteria with the high-efficiency light-emitting enzymes of fungal. In summary, these papers have now fully elucidated the molecular basis of biological autonomous luminescence through the study of both lux and loose systems. Specifically, deep biochemical studies on enzymes like MCH3H have revealed how nature saves energy through substrate gating mechanisms and proved the feasibility of engineering these enzymes. Looking ahead, with the convergence of AI-assisted design and metabolic engineering, the next generation of autonomous bioluminescence systems will be brighter, more colorful, and become indispensable living light sources for life science research. That's all. Thanks for your listening.